Well, the title that I was sort of given, in a sense, is What Do the Genes Really Say? To a certain extent, it's a little bit restrictive, and I'll explain that in a moment. But I do want to give apologies, because like Mike, first and foremost, I'm a biochemist. I've been a biochemist now for over 40 years. You can see the comparison here with the grey hair, the bald head, and the glasses, and the beard. You know, maybe all biochemists are like that, and I'm not sure if that's true or not, but I am. And really, as Mike has shown in his book, and as I enjoy speaking, the arguments, really the credible arguments, are very technical. And I don't apologise for that, although it's under the heading apologies, because I want you to appreciate my doubts as a biochemist about Darwinism. So the title is What Do the Genes Really Say? And as I say, it's a little bit restrictive, the title, although it's a bit catchy. But I also feel I want to say something about chromosomal architecture, because it's really here that we can have great reservations about whether Darwinism could possibly work at all. So I want to look at protein coding DNA, which is really what genes are. So we can see here that there's some restriction in the title. I want to consider briefly really one of the most exciting areas in biochemistry today, which is looking at the non-protein coding regions of DNA, which were once called junk, once. But I do want to look at chromosomal integrity, and I want to say and consider some of the implications. Well, this was a very important day in the history of molecular genetics, both in the journal Nature and in Science. The draft of the human genome was published. How did they do it? Well, they extracted DNA from all of the 23 pairs of chromosomes in the human. It was extracted, it was fragmented, enormously labour-intense work, took many years, several groups working together. And those small fragments were sequenced, and then the whole thing was put back together. So what did we get? Well, we got something like this. A, G, C, T, and so on and so on. And we discovered that each human cell has something like 3 billion of those 4 different letters. Well, we're thinking in terms of language, but of course each of those letters is a nucleotide base in a sequence. What is it? It's coded information. Very interesting to listen to Mike in his talk, because all the mutations that have been identified, most of which are deleterious, some of which perhaps are beneficial, but all of those mutations inevitably involve a loss of biological information. That's what we have in our DNA. Coded information, 2 important words, code and information. See, DNA can be thought of in simple terms as the code of life, and you see something here of the double helix. Again, thinking in terms of a language, yes, only 4 letters. The A and the T and the G and the C are shown there, and the words or the names of the nucleotide bases for which they refer are shown there. But we also know something of the wonder of the structure of DNA, the double helix, where there's specific pairing of those letters across the helix. Adenine binds to thymine. In terms of RNA, the T is taken away, and U for uracil is put in its place. That's one of the differences between DNA and RNA. Guanine binds to cytosine, and here you can see on the screen, you can see that pairing. And this is Watson and Crick and what they discovered on the 28th of February 1953. Well, what is it? It's a genetic textbook. Well, if there are 3 billion letters in all the chromosomes in a cell, in a human cell, then the DNA in a single chromosome has to be seen in terms of millions of those letters long. The sequence is not at all random. But the fact is this, when we're thinking of what do the genes really say, it is true to say that only about 2% of this information comprises something like 22,000 genes, although that number is being revised and revised and revised, and we'll talk about that a bit later. Very simplistic, this is, very simplistic. One gene codes for one protein. It's not really true. Mike might be grimacing at this point because it's a very simplistic way of looking at it. But the translation of DNA, and again we're thinking in terms of language, so translation is the word that is used, but it's an appropriate word. It's extremely complex. It involves a lot of chemical energy. Interestingly, it involves hundreds of protein machines, and it also includes another molecule which we've already referred to, which is RNA. Here's a fantastic little video of translation.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
These animations are freely available on the web, and with the appropriate acknowledgement you can use them in your talks, and I find them quite wonderful as a biochemist. Translation of DNA, very complex, lots of chemical energy, hundreds of protein machines, but we're moving from a 4-letter language of DNA to a 20-letter language, if you like, protein structure, mainly 20 different amino acids within protein structure. Interesting to consider a chicken and an egg situation. All living processes require complex protein machines. This is nanotechnology. Interestingly, the genetic code has the information for the production of these complex protein machines, but the genetic code cannot be translated without hundreds of complex protein machines, the information for which is contained within the DNA. So what came first? The genetic code or the complex protein machines? You see, coded information in DNA, the information has no meaning unless it can be translated. Information, אינפורמציה. אני לא מדבר עברית טוב, אבל קארול, אישה שלי, ואני גרים בערד, בישראל. אנחנו לומדים עברית. אנחנו רוצים לדבר עברית טוב. One day. Now, I'm telling you some really interesting information about me and Carol. That's true. It's important information, but if it can't be translated, you don't understand it. It has no meaning unless it can be translated. So something to ponder then. The information has no meaning unless it can be translated, and it is interesting to recognize that the 4 letters of the language of RNA or DNA do not contain information per se. They are the conveyors of information. In the case of genes, that information is ultimately going to be translated into protein structure. DNA is the perfect vector of information. Information itself is not material. So where does biological information come from? And of course that leads me to show you this book, The Signature in the Cell, written by Steve Meyer. Really a good read. You see, Darwinism has nothing to say about the origin of biological information, and it is one of the greatest evidences for intelligent design. So these are the protein coding genes, but as I say, really only comprise something like 2% of the total genome. What about the rest? Is it junk? In days gone by, it was once called junk DNA, relics of an evolutionary past. But it's not possible to suggest such a thing today. So I want to go on to non-protein coding DNA, and one of the things that is startlingly interesting and was a great surprise to biologists is that the vast majority of the genome might be transcribed. Here's another word. We'll come to understand what that means in a moment. I suppose it began through what is known as the ENCODE project that was published in June of 2007. And now we talk about not just the genome, but the transcriptome. And there are now what are understood to be non-protein coding RNAs. And there is, surprisingly, persuasive transcription. Here's a paper that was published in 2009, which illustrates the point. Persuasive transcription of the eukaryotic genome, functional indices and conceptual implications. Here's some interesting words to highlight. The majority of the genome is transcribed, and this, again, surprising, it's a lovely word to see in the text. Observation challenges many assumptions about the genetic programming of higher organisms and how information is stored and organized within the genome. This is much more than just the genes. This is what transcription is.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the Tata box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the Tata box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach. These complexes prepare the DNA for the successful binding of RNA polymerase. Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now, energy must be added to the system for transcription to begin. This energy is provided by the reduction of ATP into ADP and PI. RNA polymerase then synthesizes an RNA template from the strand of DNA. Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates and the newly formed strand of RNA is released.
The website. Fantastic. I love this picture. You know, what we have here are some of the protein machines that are involved in transcription, the transcribing of an messenger RNA from a DNA template. You can see there's lots of them. I mean, if we highlight some regions, you see the coding region is at the far end here. Then we've got all these regions of DNA that are involved in the initiation of transcription. There's the RNA polymerase itself. Here's a protein that binds to this Tata box, T-A-T-A, those letters. You need activators and in some cases repressor proteins. These are all protein machines that are all needed. What could you say? Well, you might say that this is irreducibly complex, although I'm sure that there will be those that would object to that point. Anyway, something to think about. See, the protein coding genes, in a sense, just produce the nuts and the bolts of the protein machinery of life. And so it's not surprising that much, although not all, of that protein machinery is common between species. This is why this whole concept that we have 99% of the same DNA as a chimpanzee, which is total nonsense. It's a total myth. It's a carryover from the days of sequencing proteins. And much, but not all, of our protein machinery is common between species. Why? Well, hemoglobin is hemoglobin. It's a molecular lung. It's not going to be very different between a chimpanzee and a human because it does a particular thing that both species require. But what is different is the variation of gene expression, which is very different between species. And it involves very specific transcription factors and control mechanisms, some of which are totally species specific. Interesting point. was a big surprise when the chimpanzee genome was published that certain proteins were very, very different. And a class of those proteins were the transcription factors that you saw on that animation sequence. Very interesting. It's not all. Here's a fantastic paper published on October the 20th, 2010, talking about RNA processing and export. Let's highlight some points. Nuclear processing steps require a large set of proteins, the information for which is contained within the DNA. Adding a layer of regulatory information. You see, information isn't just protein coding genes. There's layers upon layers upon layers upon layers of information. It's completely mind-blowing. So let's move on to non-coding DNA. The first thing to highlight is the fact that humans have miscellaneous unique sequences, 11%. How does that fit in with the 99% similarity with I don't know, I really don't know. But then a large proportion of our DNA and genome contains introns. What are introns? Well, have to get a little bit more complex. Here's our protein coding gene in our DNA. What is transcribed is a long version of the messenger RNA, which contains introns and exons. The exons are the protein coding regions. The introns divide them up into spatially separated groups. But enzymes cut out the introns and splice the exons together. And it's the processed messenger RNA with its poly-A tail, which I'll refer to again later, which is used to make the protein. Last one of these. Splicing. Fantastic process.
The following animation will describe the process of RNA splicing, an important step in creating the mRNA that is involved in protein synthesis via the process of translation. Key factors in this process include RNA, possessing introns and exons, and the spliceosome. Here we see an RNA molecule with a single intron. Several signals exist within the intron that are used in the splicing process. From the 5' end of the intron, these are GU, the A branch site, a pyrimidine-rich region, and the 3' AG. The GU and AG sequences define the beginning and end of the intron. Splicing is mediated by the spliceosome, which consists of several protein RNA complexes. The first step involves two complexes that bind near the GU sequence. The RNA is then looped, and 3 other protein RNA complexes bind. This final complex then undergoes a conformational change. Introns are non-coding RNA sequences that must be removed before translation. The process of removing the intron is called splicing. The intron is then cleaved at the 5' GU sequence and forms a lariat at the A branch site. The 3' end of the intron is next cleaved at the AG sequence, and the 2 exons are ligated together. As the spliced mRNA is released from the spliceosome, the intron debranches and is then degraded.
Right. Now, the purpose of showing that, and I know it's technical, is that there's another layer of information which is now called the splicing code. And here's a paper in Nature that was published this May, deciphering the splicing code. Let's highlight some things. Hundreds of RNA features to predict tissue-dependent changes in alternative splicing for thousands of exons. The code determines new classes of splicing patterns, identifies distinct regulatory programs in different tissues, and identifies mutation-verified regulatory sequences. Unexpectedly. It's a nice word that I like. Large combination of features, appearance of features deeper into introns than previously appreciated. We're learning more of the complexity of the genome. So what do introns do? They're now seen to be a code within a code. They're involved in the control of gene expression, which is both tissue and time specific. A code detected a class of exons whose inclusion silences expression in adult tissues by activating nonsense-mediated messenger RNA decay, but whose exclusion promotes expression during embryogenesis. We'll look at that again a bit later, just briefly. Pseudogenes, thought to be relics of our evolutionary past. Here's our protein coding gene sequence, the DNA, transcription, processing of RNA, translation into protein. Well, there are two types of pseudogene. There you see our genomic DNA, which can be duplicated to produce a duplicated pseudogene, which is unprocessed by gene duplication. And there are characteristic mutations in this sequence that sometimes leave this pseudogene non-functional in that it doesn't produce protein. But the fact is this, many functional genes are actually present in multiple copies, some of which become non-functional in terms of protein production. The second type of pseudogene, here you see our transcription and our processing of RNA, and the thought is that that RNA, through reverse transcription and mutation, produces a processed pseudogene that can be transcribed, but it doesn't produce protein. We think that it comes via this process because this DNA, which is the processed pseudogene, has some characteristics of messenger RNA, including the poly A tail. What does it do? Does it do anything? Or is it a relic of our evolutionary past? Well, here's something to consider. In the light that the vast majority of the genome may be transcribed into RNA, actually is the term pseudogene a misnomer? And it's my question, is adherence to Darwinian theory and dogma actually inhibiting an understanding of genomic structure and function by this wholesale determination to see pseudogenes as relics of an evolutionary past? Just very quickly give you a case study. And this is a prediction based on design. All right, let's consider vitamin C biosynthesis. The fact is humans and primates and lots of other animals, including blackbirds, can't synthesize ascorbic acid because the final gene coding for the final enzyme in the biosynthesis of ascorbic acid is non-functional. And here you see that enzyme which doesn't work in this biosynthesis of ascorbic acid. And there have been many papers over the years. A group in Japan, Nishikimi's group, demonstrated that the human pseudogene of this final enzyme, which is called GULO, there's only these exons, or the thought is that only these exons remain within the human genome, whereas this rat shows all of the exons, which are numbered, which are all there, and the rat has a functional gene for GULO, which means that the rat can synthesize throughout adult life vitamin C. But you see, is this fragment all that there is? Actually not. We've discovered that the human genome actually has all the exons. They're distributed across the human genome. Nishikimi never looked for that because his fundamental belief was that this enzyme, as a pseudogene, was a relic of an evolutionary past. But actually, the enzyme is there in its totality, although fragmented. Does it speak of physiology changes? Is there any evidence that humans can actually make vitamin C? Dennis Alexander gets really mad with me for showing this paper, and it's in a book that was published called Should Christians Embrace Evolution, if you wanted to read about it in greater detail. There are some many old papers that seem to indicate that before birth and in neonates, humans can synthesize vitamin C. Here's an old paper. One point which has been particularly impressed upon us is that though in all other respects the various stigmata of malnutrition and undernutrition are such as would be expected from an inadequate diet, infantile scurvy does not apparently occur, even when the mother is totally malnourished in terms of vitamin C. It's as though the fetus can synthesize vitamin C. And this is what they say, yet the source of this vitamin is puzzling, and the only alternative is to postulate an endogenous production. Now, I'm stepping out on a limb here because I'm waiting for the confirmation papers to be published on this. But it means, is it Darwin or design that is allowing us to make predictions which can be supported by scientific evidence? We are challenged that we're not scientists at all. Ascorbic acid in fetal human brain is far higher than could possibly be expected on the basis of maternal intake of vitamin C. Really interesting stuff. The significance of this phenomenon is unknown. Something to ponder. If it can be demonstrated that humans can synthesize vitamin C in utero, all right, I'm stepping out on a limb here, folks. I'm wanting to make a scientific prediction on the basis of a design argument here, okay? The existence of a pseudogene may say something far more about physiology and the switching on and the switching off of genes throughout life than whether it could possibly be a fossilized relic of an evolutionary past, which people like Dennis Alexander and Kenneth Miller would want to maintain. It might well then become a useful model for the understanding of genomic function and control. Life is complicated. In any cell, the vast majority of the genome must be silenced. It must be silenced. It's true in a stem cell, and it's true in a differentiated cell. The vast majority of the genome that contains all of this information, it must be silenced. The process of cellular differentiation is highly ordered. And here you see our stem cell, from which has all the information, but from that stem cell we can produce blood cells, cardiac muscle, or neural cells where genes are switched on, some genes are switched off. And this, of course, is the science of what is known as epigenetics. Really current, really interesting stuff. in any cell. The vast majority of the genome is potentially transcriptionally active, but the vast majority of the genome must be transcriptionally silenced. And here you see one of the ways in which that's done. Here's the RNA, which has been transcribed from what is known as the sense strand of the gene. Here is the RNA, which is transcribed from the antisense strand, the complementary strand of the pseudogene. Ah, pseudogenes can have function, can they? Oh, right. Producing this complex. This is broken up using this enzyme called DISA, small inhibitory RNA, which produces this released complex, which is separated. That's what RIS complex means, RNA-induced silencing complex. And that little fragment of RNA, which is transcribed, blocks the translation of the messenger RNA. And ultimately that RNA is broken down into small fragments and degraded. And that's one of the ways in which protein biosynthesis and gene expression is reduced. The stem cell genome. Stem cell genome, it's like a brand new computer hard drive. The genes are like individual programs. They may become fragmented by, I believe, complex and highly ordered processes. Vitamin C production may be involved in this particular process. Cellular differentiation, genes will be switched on and off. Transposable sequences may be involved in gene switching. These are the mobile genomic elements. And through alternative splicing, there is the creation of new gene sequences. Gene switching may well involve the introduction of specific stop codons and frame shifts in order to deactivate those genes. But this has to be a highly ordered and controlled process. And information is four-dimensional because it is expressed differently over time. Layers and layers and layers of complex information involving time. So what can we say about the blueprint of life? The DNA sequence itself can be considered as a one-dimensional code, but genomic structure itself can be considered as a four-dimensional code, because from the fertilized egg we get the development of the child over 9 months, the blueprint of life itself. So what do the genes really say? Every academic paper that is being published today, which is speaking of the complexity of the genome, in my opinion, is driving nails into the coffin of Darwinism. To the point that in time, and the change in the scientific community with respect to this, we're involved something like a paradigm shift. It's going to take time, but I believe that before too long, the scientific community as a whole will recognize that Darwinism, as described by Michael, random mutation, natural selection, is untenable from a scientific point of view. And I would go as far as to say is that actually common descent itself is thrown totally into question. Of course, within the genome there is the capability of variation, but it's within very well-defined limits. And chromosomal integrity and architecture is all to do with the limits and the edge, in my opinion, of evolution, and the edge of common descent itself. Of course, that's what the science is actually going to show in due course, but it has enormous implications for a re-evaluation for how life has come to be as we have come to observe it. All right, take a journey into the heart of the cell. Let's look at the chromosomes. Here's a lovely picture of a dividing chromosome, condensed and dividing. I just want to point out one feature of it, although there are other features, and it has bearing on the question that was over here about chromosomal fusion, and I could talk about that in the questions and answers if you want. The centromere, which is this constriction between the dividing chromosome, which the position of which is absolutely essential for the functioning of cell division and so on. I think myself is that chromosomal structure is the wonder of architectural achievement in the universe. I think so. It involves coils and coils and coils of DNA wrapped around proteins called histones, the unit of which is called the nucleosome. There's your DNA. This is a lovely picture of our negatively charged sugar phosphate backbone of DNA. That's negatively charged, right? Wrapped around maybe up to 8 different proteins, positively charged histone proteins. That's quite significant because there's another layer of information processing which is called the histone code. Because if you chemically modify the histone protein by making it less positively charged, even negatively charged in places, the negatively charged sugar phosphate backbone of DNA begins to unwind because it's no longer positive-negative interaction. It's called the histone code. It's another layer of information processing. This is amazing engineering, and cell division requires the copying of all the DNA with incredible accuracy. And here you see a plant cell, actually. And you see the chromosomes in the process of cell division. Now, this type of cell division involves the same number of chromosomes in the daughter cells as in the parent. This is standard cell division, which is called mitosis. Amazing engineering. Now, this DNA is unwound with incredible speed. And it doesn't get tangled. Copied with miraculous fidelity. And this was the science journal where the human genome was actually published, the American version, that is. A separate paper, Human DNA Repair Genes, 130 known human DNA repair genes. There's so much molecular machinery in the cell that's involved in DNA repair. It's as though the cell is doing everything it can to maintain chromosomal integrity. And since then, there have been hundreds more protein machines discovered that are involved in DNA repair. There's extensive correction mechanisms for any error. Some were anticipated in advance, which is amazing to me. And it appears that it's vital to maintain the total integrity of the DNA. Something to think about then. This is really current scientific work. Any disruption to chromosomal integrity inevitably leads to disease and death. And the current academic literature is filled with examples of this. Errors in DNA repair inevitably lead to disease and death. And all living organisms have complex protein machines that maintain the total integrity of chromosomes. And this, to my mind, is the killer when it comes to Darwinism from a scientific point of view. Profound implications for sexual reproduction, because this involves a different type of cell division, which is called meiosis. It's the formation of sperm and egg. Each sperm and each egg have half the number of chromosomes, 23 pairs in an adult cell, but 23 individual chromosomes in sperm, 23 individual chromosomes in the egg and the sperm. Half the number. Of course, we can sort of understand why that is, because at fertilization, the full complement, the 23 pairs, are re-established. The individual chromosomes in sperm and egg combine together to produce the 23 pairs of chromosomes in the fertilized egg. Just to show these simple animations, the first stage of meiosis, the formation of sperm and egg, involves the pairing of homologous chromosomes. That, remember, 23 pairs, we've got a pair of each chromosome, with the exception of men who have an X and a Y. That's our 23rd set of chromosomes, and in the man they're unpaired. That's an interesting thought. But all of the other 22 pairs, there are 2 of them, and they pair up at the beginning of meiosis. The pairing of chromosomes, well, there are 2 copies, one originally derived from the father, one originally derived from the mother. Meiosis cannot start, folks. It cannot start unless the pairs are zipped together, the homologous pairs of chromosomes. Of course, in men, it's a bit different because we've got an X and a Y, but they're paired together end to end. Now, this process of pairing of homologous chromosomes is called synapsis. It's extremely complex and very accurate. And the centromere is essential in that pairing process. You cannot haphazardly move the centromere wherever you like and hope that you're going to get the pairing of homologous chromosomes. How do I know that? Well, I know it from a scientific point of view. Emerging roles for centromeres in meiosis 1 chromosome segregation. This is Nature Genetics, December 2008. It's essential and conserved. We're beginning to understand the numerous roles that centromeres have in chromosome segregation. Fantastic picture of the pairing of those chromosomes. They're in the process of division already, but they're being zipped together. Some of those proteins are called zip proteins. What is being produced is a complex called the synaptonemal complex, which is an incredible structure that's involved in meiosis. And here's another picture of it, which is taken from a recent review of genetics, March 2009. Look at the title, folks. Look at the title. The Consequences of Synapsis for Mammalian Meiosis. Is this just an abstract idea, what we're talking about here, or is it fundamental to this whole argument of design or Darwin? It has been known for decades that errors in chromosome synapsis, that is, asynapsis, are associated with impaired fertility, with high levels of asynapsis. Sterility is seen in both sexes. The second part of meiosis is similarly important, the recombination of DNA when material is being passed from one chromosome to another. Here's a fantastic animation sequence. Now, if you look very closely, you will see this process of recombination where material from one chromosome is being passed and shared with another chromosome. But here you're getting specific base pairing taking place. You can't haphazardly alter that for this process to continue. Chromosome recombination requires total and complete alignment of homologous chromosomes, and as we've understood, the position of the centromere is fundamental to this process, and it involves a careful check on the integrity of the DNA sequences at the recombination hotspots. It's got to be correct. It's vital to maintain the integrity of the chromosome. There are hundreds of protein machines involved in maintaining the integrity of the chromosome. It seems to be a fundamental of nature. So there's something to ponder. Follow the argument. Meiosis cannot begin without synapsis and recombination. It can't begin without synapsis and recombination. Synapsis cannot happen without the maintenance of chromosomal integrity. It can't. If there's no meiosis, there's no sperm or egg. If there is no meiosis, there is no survival. So in conclusion then, is it actually, it's true by definition, it's a tautology, the survival of the fittest? I think not. More accurate to say, the survival of the fertile. And the maintenance of fertility requires the maintenance of chromosomal integrity. And I would submit to you this: the maintenance of fertility and Darwinism are totally incompatible. Doesn't have a mechanism, folks. Of course there's variation, but there's a limit to common descent that will be measurable in terms of genomics in due course. We've looked at all of these things. What does the science really say? What do you say? All right, that's it.
Thank you.